Note to participants before we begin. All sessions will be available in English or French. To activate translation services, you must set your language preference in Zoom. Today's session will be entirely in English. Therefore, English-speaking participants will not need to activate translation. Now, our French translator will share a brief message on this channel in French to provide our French-speaking participants the necessary instructions for activating translation services.
Toutes les sessions seront disponibles en anglais ou en français. La session d'aujourd'hui sera entièrement en anglais. Pour activer les services de traduction, vous devez définir votre préférence linguistique dans Zoom. Si vous regardez actuellement cette session dans la plateforme Hoover, veuillez sélectionner Participer en Zoom. Une fois dans le Zoom, pour configurer la traduction, cliquez sur l'icône du globe terrestre en bas à droite de votre page Zoom. Sélectionnez le canal de la langue souhaitée. Si vous constatez des interférences sonores sur le canal de votre langue, essayez de sélectionner Couper le son original.
Welcome to the Feed the Future Innovation Lab for Legume Systems Research Annual Virtual Convening Day 1. Thank you for joining us. We look forward to sharing innovative approaches and discussions on legume systems research over the next 3 days. My name is Jan Fierro, Communications Manager for the lab. I will be facilitating our sessions this morning. A few details on our meeting format. You can view the session in the Hoover platform or on the Zoom platform. If you choose to participate in Zoom, please also keep this session open and available in the Hoover platform. This will allow you to participate in Q&A, chat, polls, community, and to access the session agenda, as well as these features are available in Hoover exclusively. I would also like to highlight the community feature on Hoover. Here you will find additional resources on many topics and be able to engage in ongoing conversations throughout the convening. Please note the chat feature in Zoom has been disabled. At this time, we also ask that you take a minute and click on the Polls tab on the Hoover platform and complete our poll of the day and vote for your favorite legume. Results of the poll will be shared in tomorrow's opening session. At this time, we welcome Mr. Daniel Bailey, USAID Agreement Officer Representative for the Legume Systems Innovation Lab, who will provide opening remarks.
Thank you, Jan. Thank you, everyone. Thank you to the Legume Systems Innovation Lab, which is more than just Barry and John and David, but also to everyone involved today. So thank you for taking this time out of this week to participate. I know it's been a difficult and challenging year for everyone with COVID and just the general trials and tribulations of research and trying to track everything through the lens of what the U.S. government is doing through a presidential transition. 2021 is starting out as a much better year, and as coronavirus will hopefully fade into the background by the end of the year, we look forward to getting back to more business as usual. Although on one hand, I do hope that business as usual includes more opportunities to connect virtually, like through these platforms that have been strengthened and built up over the last year. I hope that communication has improved for everyone involved in international programs such as this, and that the telecommunications in your own countries are improving on a weekly or monthly basis. A couple things I wanted to point out as we start this new year. And the first is to remind you that you know, USAID places a very strong emphasis on gender and gender analysis in everything that we do. So please make sure to continue keeping up strong gender analyses in your research and in your work. Always try to remember, you know, how does this, whatever you're working on, impact men and women differently? How does it impact youth differently? Are there other often marginalized people groups that could use this work differently? Similarly, local capacity development, as we are entering I believe the third year of this activity, you know, what needs to be done to strengthen the local capacity where you are with the people who you work with? Is there something within the opportunities within Legume Systems Innovation Lab to improve those local capacities? Unfortunately, the Legume Systems Innovation Lab doesn't have the resources to improve every aspect of local capacity development, but there may be simple things that we can do, even just making sure people are connected with one another. And then finally, missions, USAID missions. Please always remember to keep them updated. Always run any public events, especially when there may be press, whether invited or uninvited. Make sure that those are passed along to Barry and John and Dave in the ME, and that we get approval beforehand from the local mission. And please also remember to get approval before meeting with local government officials, including, you know, research heads at national research agencies, especially ministries of agriculture. Sometimes these things happen coincidentally to other work you're doing. If you are asked to give an impromptu brief on what you're doing within a research agency, please report that immediately to the Innovation Lab so that we can report it to the mission. And finally, over the next year, you may hear about a product lifecycle management tool and a product profile management tool. I just wanted to put this on your radar to make sure that you are aware that USAID is starting to develop and to implement these tools as part of our core management processes of research research programs. And know that this will be a very important management tool as we move forward. And with that, I thank you again for your time this morning, and I turn it back over to Jan.
Thank you, Daniel. We appreciate your words and your insights, and thank you for sharing on some new and exciting things it sounds like that are coming down the pipeline with USAID. Next to share a few welcoming words is Dr. Doug Buehler, Director of Ag Bio Research and Assistant Vice President of Research at Michigan State University. Doug?
Good morning, everyone, and please accept a welcome on behalf of the It is a great pleasure to be here today. And I can tell you that for those of you who are from a more tropical climate, being in Michigan this week will be quite an incredible experience for you. The middle part of our country is in the middle of a very strong storm, and so I hope all of you that are residents here in the United States are also safe in handling our challenging weather in a safe way. It was for me earlier to watch the slideshow from last year's convening, really kind of an eye-opening event to just think about what has happened and what we've all been through in the last year. I think I would bet for many of us it was maybe one of our last significant trips, certainly international trips, before the world was locked down due to COVID. And to think about how we have come together and been able to maintain our work and move forward in ways that I don't think any of us ever dreamed that we could have done less than a year ago is really just quite heartening. And it does show really the resiliency and the innovation that we can bring together to do our work even under some of the most difficult circumstances. So for me, having an opportunity to make a few comments to this group is always a real pleasure. This laboratory and this activity has been a really important component and really a flagship program of Michigan State University for a long, long time. International activity and research around legumes are two very important issues for us, and will remain so for a long, long period of time. We really see this as a core of what we do, and as something that, again, the international partnerships, the connections that we've built, the students, and all of the things that we've been able to do are just fantastic, and we only see more and more of this as we move into the future. So when I look at this laboratory, again, having been involved with it for a number of years and in some different capacities during my time at MSU, it really, to me, is an incredible example of how we start with some very fundamental science, whether it's genetics and nutrition and other things. We conduct solid, strong research, and we take it all the way to application, not only domestically, but around the world. And that's really, really, I think, is emblematic of the mission of land-grant universities, many of the research institutes and universities represented here, certainly USAID and many other institutions. And so that's really a great thing to see continue to happen and to continue to move forward at times where this type of work is very important. One thing I do want to mention, and again, this is just in a way to reiterate the support and the commitment of Michigan State University to this laboratory. Many of you may be aware that Barry Pittendry has taken a very, very wonderful opportunity at Purdue University, and will be at least technically leaving Michigan State University. We're sorry, very sorry to see Barry leave, but we also know that it was an incredible opportunity, and say also an important way for us to strengthen our partnerships with Purdue. The good news for the laboratory is that we've negotiated an agreement with Purdue University so that Barry will continue to be the lab director with really no change in the way things have been happening. Barry is already technically an employee of Purdue, and I think we've kept everything pretty much running as usual. So we've been working behind the scenes here at Michigan State with Barry and John and the rest of the staff to put things together that will keep things running very smoothly, and again, maintain our commitment. And as Barry said, he's committed to this activity, and we'll continue to work with him and the rest of the staff to keep everything running as smoothly as possible. So I also want to just take a moment to thank all of the folks in the management office and all of you again for the excellent work that you do. And again, having done it under some very, very trying circumstances. I believe we've learned some things that we're going to continue to apply that will make us more efficient and to get some of our work done. But I also, as I think all of you, look greatly forward to the time that we can actually get to work together again and to meet and to exchange ideas in more informal settings. One of the observations that I've made of working, I don't even want to think about how many hours of Zoom I've been on, or any of you have been on now in the last 11 months. It's a very effective tool. It can allow us to do a lot of things, but I do believe that for my money, it's a very transactional way to do business. So I find that people that I already have an existing relationship with, we do very well. But I can't help but wonder, with the meetings that we've not had and the interactions we've not had, how many how many people have you not met that you might have met more normally with normal activity? The people that you meet in breaks, happen to sit next to it at meals and things like that happen at convenings like this are something that we've lost. And so hopefully we'll get back to that. We can blend the good things that we've learned and the great power of these tools of virtual meetings back with the ability to get together and to really think about things and come up with new ideas and to build relationships. So let me just wrap up my few comments, again, saying that it's a great pleasure for Michigan State University to be part of this. Again, this very much aligns with what we do. And this university has had a long commitment and really was, I think, one of the first land-grant universities to be very committed to this type of work. And we now have a 50, 60 year or more history on some of these things. And I hope to think that it's just the beginning of the things that we're going to do and can do together to meet the important goals of feeding the world in sustainable ways. We all know that we have some very severe challenges in front of us, certainly climate change and the need to develop more resilient food systems. All of these types of things will continue to be there. Our work has never been more important. And so as we look forward to building on what we have, the issues around gender and equality that we all know are so incredibly important, how do we blend that with our science and through our partnerships to move forward is really a great challenge for all of us. And I do believe that this group is very well suited to meet those challenges. And I look forward to playing some small role in helping to make that happen. So again, I hope that you're all staying safe and that we have a great meeting. And I'm looking forward to joining in on as many sessions as possible. Thank you very much.
Thank you, Dr. Buehler. Next, Legume Systems Innovation Lab Director, Dr. Barry Pittendry, will share insights and objectives for the next few days.
So great, thank you very much. And I'd just like to thank Doug and Daniel for their opening remarks. And I'd just like to echo many of their comments that we strongly appreciate everybody's resilience through the last year. I'd like to speak to this a little more. First and foremost, I really want to thank USAID and their team for all their support since the inception of this lab, but certainly over this last year, as we're going through a considerable amount of adaptations at every single level. I would also like to thank Doug Buehler and MSU for their ongoing support. Doug and his team have been incredibly supportive throughout this process of everything from the day we decided to compete for the management entity and every day afterwards in terms of management and adaptation and the necessary support that we've had to move this forward. So their efforts are extremely appreciated, and I appreciate Doug taking time out this morning to also present, and it just shows the ongoing commitment of his team and Michigan State University to this program. I do want to just thank the management office team before we get into the details today of the overall program. It's been an incredible set of people that we've been able to work with since the inception of this program, and I'd like to thank them for all their efforts over the last year, especially in all the challenging times that have been going on, and all the challenges associated with getting this meeting together over the next 3 days. They've done an incredible job to collaborate and adapt in this management process. But I definitely want to reach out and thank all of our researchers for all of their efforts since the inception of this program, but definitely over this last year. We realize it's been a challenging year for everyone. We appreciate all your efforts. It's been a challenge for people both professionally and for many people on personal fronts. And we realize that in spite of all these challenges, many or all of you have really risen to the occasion and innovated and come to the table with new approaches of doing business that have really helped us to move things forward. And I think it's incredibly important within the context of COVID-19 because As we all realize, legumes are an incredibly important part of resilience for people globally, and especially the global poor. It's an important food source, protein source, as well as micronutrient source for many people globally. It's a protein source that can be stored, and as we saw the rising prices of legumes globally this past year, how much people depend on it in all social economic strata. So the work that you're doing is going to be very important as we go through the next phases of this crisis, and as we enter into issues of global climate change. A lot of the research that's being developed by teams in this program will ultimately have ramifications for many people in the coming years or through multiple generations. So the work you're doing is incredibly important and has real long-term potential for impact. So I'd like to thank you for sort of working with our team over the past year to adapt to sort of our new realities. And I would like to echo Doug's comments that we're hoping that at some point we can return to those at least partial in-person interactions that are so critical. But we all have had to survive through numerous Zoom calls through this past year. But I think you know, it's been we've been able to work very effectively as sets of teams towards these issues. So I would like to jump into a little bit more details about what we hope to accomplish over the coming days. And I was going to provide a list and give you a little bit of background on what we're looking to focus on. First, we definitely appreciate the research projects that will be giving presentations over the coming days. We'll be talking towards cross-project learning and exchange. We'll be bringing in our Technical Management Advisory Committee in to see progress and to interact with PIs. They're a really important part of this team, and they're an important part of the team that I would really like to place a strong emphasis on the thanks for all the work that they do. They are really a behind-the-scenes team that has done so much to help us move forward, and they've invested at incredible numbers of their hours on a volunteer level and incredible levels of their expertise to help drive this program forward. From everything from review of concept notes to review of programs to constantly working with us to help projects move forward and maintain the highest quality of academic rigor. So I really would like to shout out to the TMAC. I'd like to especially mention Jeff Alwang and Bob Easter, who played roles as a chairperson for the TMAC, and they've invested an incredible number of hours in the work that we've been doing, and I'd like to thank them. This is a task that absorbs a lot of their time, a lot of their expertise, and we're really fortunate for the TMAC to have globally recognized actors and people that are at the top of their profession that are providing insights and efforts to help us move this forward and move this program forward. It's without the TMAC, we could not function. So I'd like to really emphasize the efforts that they brought to the table. We would like to highlight the research of external stakeholders and for possible scaling partners. As Daniel mentioned earlier, one of the long-term goals of all of these programs is to scale. Our goal is for research towards scaling. It's to provide those innovations that ultimately allow others to scale content and to take what we're doing forward into new systems, and to take that out into the hands of the people that need it the most. We'll also review and update the strategy of our white paper. There's an ongoing living document that we're continually working on in order to bring out the concepts of where the community is and where the community needs to go. And we feel this is a very important living document that we will continually update throughout the process, or throughout the time period of our innovation lab. Our RRT program will also present their work for transparency to PIs. They work very closely with many of the PIs to help us deal with a whole series of issues and help the PIs move forward their projects towards greater impact. And it helps towards highlighting the work of USAID. And we hope that it helps the PIs towards launching their next rounds of funding and opportunities that may go beyond our particular program. So we'll also go over and talk to over the coming days about administration. How do we properly and effectively communicate with USAID missions? The missions are our advocates in the country. They are real potential opportunity partner entities to help us have impact, both in the context of this program and beyond. But we have to manage these relationships carefully and build those relationships. And building relationships involves effective communication with USAID missions. So we'll talk about this issue in the coming days and why there's a real special need for us to make sure that we're building those quality relationships with our mission partners. And we will go over why there's a need for you to capture those great photos that are important for us to tell the stories. And when is it the right time for you to hire or bring in a professional photographers to get those really top-notch photos that are necessary to tell those stories. Picture's worth 1,000 words, and we need those 1,000 words to tell your story at the right place and the right time. So we encourage you to get those photos that will be so critical moving forward when we have to tell your story, and we can bring what you're doing to the forefront. We'll also talk about the EMMP and collecting documents for ROC and also purchase of pesticides, seeds, and fertilizers. These are a series of technical issues that we're going to have to deal with in order to move forward effectively. So they're details that sometimes people don't want to talk about, but there's things we have to bring to the forefront and have to be dealt with very, very effectively a priori in order to make sure that we're dealing with things properly and ethically. We'll also be having a Cowpea Atlas brainstorming session that will be help us move forward and understand how to develop a Cowpea Atlas that we think will be very important for the community, both within the context of this program and moving forward. And we'll also be talking about COVID-19 impacts moving forward, travel, students, in-person precautions, etc., and what that's ultimately going to mean looking forward. Additionally, we will be trying to do a significant amount of networking during the next several days. So this will kind of bring us forward to understand what we're hoping to accomplish over the coming days. So I know our next session will start at 10:45, and that will be brought to John Medendorf will lead that next session.
Welcome back, everyone. As Barry mentioned, my name is John Medendorp. I am the Deputy Director of the Legume Systems Research Innovation Lab, and I welcome you once again to our Research Session 1. In this session, we will hear from 3 of our research projects. The format of the session will be a pre-recorded presentation by the project team, followed by a short question and answer period. In order to submit a question, please use the question and answer feature on the right panel of your Whova convening page during the presentation. Since our question and answer time is very limited, we ask that all participants monitor the question and answer panel and upvote those questions that are of the most interest to you by clicking on the thumbs up icon next to the individual question. In the open question and answer session at the end of each presentation, we will be selecting the questions with the most upvotes for further elaboration. Also, to help us in managing your questions, please preface your question with the alphanumeric designation for the presentation. This is session 1. There will be 3 presentations. So the 3 presentations will be designated as 1A, 1B, and 1C. Please list the corresponding alphanumeric at the beginning of your question. I will provide the corresponding alphanumeric designation before each presentation. Our first presentation is System Research to Identify Legume Options for Sustainable Intensification, Pigeonpea and mung bean as test cases for scaling in West Africa. This project was an initial activity of the Legume Lab and recently completed research activity. The project was led by Dr. Sieglin Snap of Michigan State University. Presenting today for Dr. Snap's team is Dr. Princess Hayford, also affiliated with Michigan State, and the question and answer designation for this session is 1A. So preface all of your questions with the alphanumeric 1A, please. And with that, we'll go to the presentation.
Hello, everyone. I am presenting on systems research to identify legume options for sustainable intensification, Pigeonpea and mark bean as test cases for scaling in West Africa. I am Princess Hayford, and I worked on this project with Dr. Snap. The goal of the project was to test case the suitability needs for Pigeonpea and mark bean in West Africa. Why Pigeonpea and mark bean? These two crops perform unique functions in cropping systems. Pigeonpea, a long duration multi-purpose crop, is able to provide food and vegetation for fodder. And it has the ability to enhance soil nitrogen and phosphorus availability with its association with the roots biome. Mark bean, on the other hand, is an early duration crop that can be grown in relay and in intercrop systems for a hungry season food source and provides new avenue for income generation. Smallholder farmers in arid West Africa are challenged by insufficient access to land, inputs, and education. Also, food production and quality of diets are insufficient. And this may be due to the limited crops that are grown. These crops are sorghum, millet, cotton, cowpea, with few legume seeds available. Legume species options vary in growth types. Annual pulse crops are mainly grown for food and income, and they often provide modest soil fertility benefits. An example of an annual pulse crop is cowpea, as we see in this picture. Then when we look at perennial trees, they are good for soil rehabilitation with soil carbon gains and fertility, such as Gliricidia. But most often their products are few, which is limited to forage, fruits, and share nuts. Then we have the semi-perennial crops and intercrops. An example is Pigeonpea. They are often win-win because they provide food and soil benefits. Pigeonpea can be grown as an annual or as a perennial. We did examine potential for perennial Pigeonpea as a food or fodder crop in Bamako, Mali. And we realized that Pigeonpea that were ratooned produced more biomass and grain compared to first-year Pigeonpea. So when I say ratoon, then what I want to say is after the first season of cultivating Pigeonpea, the stems are cut off to leave about 30 centimeters for it to regenerate. And this regeneration results in more biomass and grain compared to the first season crop. And following expert interviews, we noted that the locally adapted Pigeonpea varieties perform well. However, there was no documentation on genetic characterization and phenotype, and this is highly recommended. We went ahead to conduct a survey on Pigeonpea research in Ghana. And from the survey, we noted that The improved Pigeonpea varieties that are being tested are not adapted to the conditions in Ghana. The local genotypes that are usually long duration are showing promising signs of growth and yield. Then the dual purpose ones, the ones that are grown for grain and forage, they have high potential. And even when their branches are pruned, they still do not reduce yield, which is very good. Then we noted yam farmers use Pigeonpea as stake. And these are often grown as a mixed crop. So in this picture, we have Pigeonpea and yam growing. So the yam vines will be climbing on the stems of Pigeonpea to support their growth. Then a major production challenge that we noted in the wet season was the free range of livestock. And this is predominantly in the northern parts of Ghana. We did a field experimentation where we evaluated Pigeonpea as sole and intercrop with sorghum. We looked at two different growth types, medium and long duration. We had 5 cropping systems: sole Pigeonpea medium duration, sole Pigeonpea long duration, sole sorghum, sorghum Pigeonpea intercrop with medium duration, then sorghum Pigeonpea intercrop with long duration. This is sorghum Pigeonpea intercrop. Then here we have a long duration Pigeonpea and a medium duration Pigeonpea. So we can see the contrast in growth of these two varieties. Following field experimentation, we did model parameterization and calibration in APSIM. And APSIM uses daily weather parameters. So these were measured on station. So we provided daily temperature, which is the maximum and the minimum, then solar radiation, then rainfall. Then we input soil parameters, then crop parameters, that is the sorghum and the Pigeonpea that were grown. Then soil nitrogen, soil phosphorus, and surface organic matter. Then from there, we proceed to calibrating the model. We use the biomass of Pigeonpea and sorghum and that of their grain yield to do the calibration. To be able to create crop coefficient, we use the phenology of the two crops to help us do that. And through the calibration, we have to make sure we get a balance between the observed and the simulated data. So we had to run the model for some time to make sure we created that balance. This figure shows the simulation of sorghum and Pigeonpea, system evaluation over space and time for crop yields and water use efficiency. In the intercrop system, we noted that there was a little suppression from Pigeonpea. However, the model was able to calibrate the system very well with their crop yield and water use efficiency. Then we simulated Pigeonpea yield over a period of 30 years under fertilized and unfertilized conditions. And the results indicated that high phosphorus soil supports high to modest grain yield in most years with no crop failures. Then the interesting part was with the intercrop Pigeonpea. Under no fertilized condition, we still had substantial yield from the intercrop system. And this is very good for smallholder farmers where access to fertilizer can be difficult. So if you have a cropping system that will not require application of fertilizer and will still give you substantial amounts of yield, then that is a very good option to go for. Then with field experimentation and modeling, we looked at the system, overall system water use efficiency. And out of the 3 sites that we worked, we noted that 2 sites gave higher water use efficiency, with the intercrop system giving the highest water use efficiency compared to either sole crop sorghum or sole crop pigeonpea. Finally, we did crop climate suitability mapping, and this was done using Google Earth Engine. And in this app, you have to provide the location of the crop phenology parameters. So in this case, we wanted to create a map for pigeonpea and mark bean. So we provided those parameters in order to be able to create that map. So this is a pigeonpea suitability niche that we created using the crop climate suitability mapping. And we realized that the northwest part of Ghana agriculture is limited by dry conditions. However, most of the agroecological zones showed favorable conditions for pigeonpea cultivation, with only few areas in the coastal areas due to the overly humid conditions there. However, this was not the case for Mali. In Mali, we realized that most of the agroecological zones seem to be unsuitable for pigeonpea production. But most of the suitability is limited to the southern part, which is good. Then we did the same for mark bean across Ghana. For mark bean, the potential for its cultivation is limited by excessive heat and moisture. Most of the agroecological zones seem to be unsuitable because of heat and moisture. However, there are few zones that indicate suitability for its cultivation. So what are the future prospects for these novel legumes in West Africa? From our studies, we can see pigeonpea is beginning to be explored as a forage shrub and co-products of soil fertility, stakes, vegetable, and grain crop. The local land races that are grown are often in mixed stands, and the seeds are readily available in the markets. For the modern varieties that come from East Africa and India, they are mostly short and medium duration and are very low yielding. There is urgent need for genetic and phenotypic characterization of these local varieties that are available. And for mark bean, there's almost entirely no research that is going on in West Africa and particularly in Ghana. However, there are few suitable niches for its cultivation in Ghana, and that needs to be explored. Thank you.
Thank you, Princess. And I would like to now invite Dr. Sieg-Snap and Dr. Princess Hayford to be available for questions. I will start out with a question of my own, which is, have there been any connections made between the West African pigeonpea production and the Indian demand markets for pigeonpea? Are there any exports from West Africa to India at this time?
Most of what we found, at least in Ghana, is very local markets. These are for home consumption. So no, I would say that really remains to be explored.
Great, thank you. And then another question is, most of this research took place in Ghana. What are your thoughts about scaling this to the region as a whole? Are there good possibilities for scaling both of these crops to the region?
Indeed. That's why we put in a proposal, which was highly ranked, as you'll recall. Unfortunately, not funded by Legume Systems Innovation Lab.
And then any insights on the willingness of farmers to adopt these crops? Is there possibilities for increasing the amount of production in West Africa?
Princess, you want to come in?
John, can you repeat your question again?
Yes, what is the willingness of farmers to adopt these crops? Is there an openness to these crops as commercial crops among the farmers, or is it mostly just a household use?
Well, farmers are open to producing Pigeonpea from the survey we conducted. But I think for the start, it will be basically for the household before, if markets can be created, then they can have it on a commercial scale. But if we can get farmers producing it for household use, that would be very good, and that would be a good start.
To get into commercial markets, there is the issue of wanting the big bold seed for commercial markets for doll purposes versus local use is much more often using types that are adapted to pest. There's a lot of pest pressure, but there wasn't any pest problems. So I think this is a really tremendous untapped potential that I hope at some point there'll be some genetic and phenotyping done perhaps with ICRISAT, which has the world's germplasm for Pigeonpea. Again, there's been some work supported by Africa Rising and very much collaborative with our work, and that we hope to eventually work with them again. Because in fact, there's huge potential, but there needs to be one of the problems is that the local varieties are very adapted, but they don't yet have, and they're pest resistant, but the improved varieties actually are not yet improved for these conditions. So there needs to be some breeding work and genetic characterization, in my view.
Thank you very much. And that concludes our question and answer session for the time being.
Voilà, avec ça, on va terminer la session de questions-réponses.
We would now like to move on to the next presentation, which is Optimized Shrub System, an innovation for improving cowpea yields and strengthening smallholder resilience in Senegal. This project is ongoing and is led by Dr. Richard Dick of Ohio.
Ce projet donc se poursuit aujourd'hui et est conduit par le docteur.
Presenting today are Dr. Richard Dick and Dr. Musa Diangar from the project team. The question and answer designation for this particular project is 1B. That's 1B. And with that, we'll go to the presentation.
Good afternoon or morning, depending on where you are in the world. My name is Richard Dick, and my co-PI Musa will be on later. We'll do a tag team type of presentation. And we're talking about a project that is involving what we call optimized shrub intercropping system as a way to remediate soils and particularly look at the potential to improve cowpea productivity. So in the Sahel in West Africa, a major constraint, ongoing ecological challenges, just the intensification of agriculture and of the landscapes for fuel wood, and a reduction in soil quality, and ultimately degrading soils is an ongoing challenge in these environments. And this is shown in this particular figure here, where you can see the one line is an exponential growth in populations in West Africa, and at the same time the lower triangle showing that the millet yield has not really increased in the blue triangles, has not really increased in the 1960s, and people are feeding themselves, but basically adding more land. And that's what's shown in the millet area here, where it's tapering off the amount of land that goes into production. So what we really have to do now is start increasing yields. And green revolution technology has not been adapted, as I'm sure most of you are well aware. So we have to look for other solutions. And one of the potential things that we've been working on for the last 20 years is the use of intercropping local native shrubs, Polystigma reticulatum and Gera senegalensis. And we've been studying these in various capacities, which I'll talk about more in a minute. But this is what they do naturally grow in farmers' fields, and these two particular species are found all through West Africa, from Senegal all the way to Chad, and they do survive in farmers' fields. So typically what happens is that there are a few hundred shrubs, 200 to 300 shrubs are fairly common from our work in Senegal, and it's probably typical throughout the Sahel. And being in farmers' fields, the farmers want to get rid of them, so they coppice them, they cut them at the soil surface, put them in a pile, and then burn them, which is shown in the right-hand corner here. And sadly, these degraded soils, this doesn't really help, because this is organic matter that could be going in the soil, which is shown as potential biomass, but they want a nice clean seed bed, and they end up then with these sort of stark moonscapes with these burn piles and carbon charcoal in these localized, but they're losing a lot of organic matter that's going up in smoke, nitrogen and nutrients and carbon and so forth. At any rate, it makes for a seed bed that they like, and then they plant the crops after that. So in our particular system, we've developed what we call an optimized system, and done a lot of research on this from soil microbiology, hydrology, agronomy, and a lot of different ways. We've published 30 plus papers on this system now in over the last 20 years, and ended up with this what we call optimized shrub intercropping. And from a long-term site, this is a picture here showing minus shrub and plus shrub, and you can see the growth response. And so in this system, instead of the 200 to 300 shrubs per hectare, or even less, depending on the farmer, we've increased it up to 1,200 to 1,300 shrubs per hectare. So if you put a shrub on every square meter, you'd have 2,400. We're roughly half of that maximum possible shrub in a hectare. So, and the second thing we're doing is we're not burning the residue, we're chopping it up and putting it back in the soil. And this is providing a fairly high amount of organic matter, especially for a semi-arid environment, where we can put 3 to 4 tons of biomass back in the soil if you get these, if you increase the number of shrubs per hectare. So the system is relatively simple to the idea. So the coppicing is still there, but instead of burning on the left-hand side here, the residue is put back in the soil. You can see the small pieces here of the recently incorporated material before the crops are planted. And this picture down below in the lower right-hand corner shows one of our participating farmers in our on-farm pilot testing of this system, planting, in this case, planting Gera seedlings in order to bring them up to the 1,200 to 1,500 shrubs per hectare density. So, as I said, we've done a lot of research on these systems, and they clearly have a real strong benefit. They take degraded soils and remediate them. Within 3 to 5 years, you can see, especially in the north and the sandy soil, you can see dramatic increases in the soil quality in terms of the organic matter, the microbiology, nutrient availability, and so forth. And and another important part from global climate change is it is a way to sequester carbon. We've doubled or even more than doubled the amount of carbon in soils after 8 to 10 years of putting these systems in place. The other really interesting thing is that we found that these shrubs do what's called bioirrigation, what we've coined as bioirrigation, which is hydraulic lift, a phenomenon driven by hydraulic lift. We published a paper actually in 2018, several papers on this, but in 2018 published a paper that confirmed that indeed water that's pumped up by shrubs from the wet subsoil, very deep, these shrubs can go 20, 30 feet deep down to get to the top of the water table. And they bring water to the surface at night when photosynthesis stops, and they leak that water out into the surrounding soil. And that's enough to actually keep a crop. We've shown that this hydraulic lift water can get a crop through a fairly long in-season drought period and keep the crop growing. And so that's a very important finding. And then the other important findings, we find that all crops get to maturity quicker. So that's another important property of this system, because in a semi-arid environment, you often have erratic rainfalls patterns. And so the sooner you get your crop in and out of the season, out of the system and to maturity, that's a real advantage in these erratic low rainfall regions. And the net effect is that we get dramatically increased yields, and the yields increase as the soils are more degraded and more sandy. And this is the site here, the Gara senegalensis site, which is where we see the most dramatic effect. And Gara does grow in the drier region, Periostigma in the wetter region of the Sahel. So the particular objectives of our project are to do on-farm pilot testing and of cowpeas. And this actually is embedded in another project we have funded through the Peanut Lab, which is really the primary funding to set up and work with these pilot farms. But within that, we're imposing some cowpea treatments. So one is, I'll talk more about this in a minute.
Minutes.
And then some of the main components that we're following here are screening of cowpea varieties, looking for varieties that are that are better adapted for intercropping with shrubs. And then we're also monitoring some of the soil properties and so forth, and then ultimately disseminating findings of the project. So just to quickly conclude my part of this, we're doing an on-farm pilot testing where we're doing plus OSS and then the traditional system shown on the left here, and and things are being monitored in soils, agronomic productivity, socioeconomics, and again that's really being funded by the Peanut Project. But with the same farmers, we were planning to take 12 of these farmers and see if we could do a sequential 2 crops of cowpea in sequence. And the idea here is that with the OSS, really providing a better soil and hydraulic lift, that would be a way to perhaps allow that second crop to finish after the rains had stopped. And so we wanted to present, we were supposed to do that last summer, but due to COVID and just the inability to get technicians out and with the lockdown, we were unable to do that. So that part's been postponed until next summer. But we did do the cowpea variety trial. And with that, I'll turn it over to Musa Dianga, who will go over the results we have so far. And we, as he mentioned, we had a major infestation of insects this particular year, which caused some issues in terms of what we can measure. So with that, I'll turn it over to Musa.
Okay, thank you very much. I'm a cowpea breeder at ISRA, and I'm involved in this project in the objective 2, which is the screening of the cowpea cultivar for superior performance on the OSS. So if you look at this picture, you can notice down where it's written no shrub plots compared to where it's written plus shrub plots. So what you see is an increase of the biomass. So we have one variety composed of 4 lines. The picture is not really good, but still you can see the difference. So that is showing that the shrub is increasing the yield or the performance of any variety that you have. Next slide. Yeah, we can go deeper. If you look at this box plot, you have in the left Pigeonpea reticulatum, and you have in the right Gara senegalensis. So Pigeonpea was in the south at Nyoro, and Gara senegalensis was at Chess. As you noticed, the traditional management system and the optimized system are very different. The optimized system always outperform the traditional system when it's about Pileostigma reticulatum. But we see that the performance, no matter the management system, when it involves Guerra senegalensis, you see it's consistent. So as a preliminary result, we can assume that Pileostigma system is more suitable to the variety of the copy that we have used. So when you compare the two, what we have obtained in the south, Nyoro, and what we have obtained in Chess, you can see that Nyoro outperform Chess. Next slide. This is due to many factors, but here, when you consider the diameter of the cullet, which is showing in an extent the vigor of the plant, we still notice the same trend. So shrub versus non-shrub. So we have better performances in shrubs, especially when it's about Pileostigma reticulatum. So in general, you can notice that in Nyoro we have better performances. In Chess, we don't have very good performances. But this is also due to the fact that the rainy season at Nyoro is always better when you compare it to the rainy season at Bombay. Something else also is the presence of the insects. We have noticed during this trial that we had an important rate of infestation of Amsakta and the thrips. So this is the reason why we can't show the results of the grain yield, but still we have obtained very good data considering the biomass, the diameter of the cullet. Next. So for the fresh biomass, so we have taken it in the basis of 16 plants and still the same train. The results are consistent in Chess, where it involves Guerra senegalensis and the traditional management system. But when you compare it to the traditional management system found in Nyoro with the Pileostigma reticulatum, that is always the same trend. So Pileostigma reticulatum outperform way better the Guerra senegalensis. Next. And here, this is some information about the abortion that we have noticed. So because of the COVID situation, it was very difficult to have a student there, and knowing also that we cannot be there all the time. So we experienced some plant abortion. And we have took some measurement on the plants that we had sampled. And as you can notice, the results were pretty much consistent. So that means that either in the plots in Chess and in Nyoro, we had experienced plant abortion. So if you consider this as a covariate, then if you consider the two locations, there is no significant differences. Next. This gives us more information. You can focus on the interaction of location and genotype and system and genotype. What you can see is we only have significant effect when you consider dry biomass variate and plant abortion. For the rest, we did not have any significant effect when you consider the interaction. But when you take the main effect, which is the management system, the genotype, and the location, in the location you can see that there is a significant difference in all the variables. And that is the same thing for management system. However, the genotype, they perform consistently in all locations. Next. Also, when you see in this slide, you have the performances. Kerndari is at Nyoro, Kermatar is at Chess, and you always see that Kerndari outperform Chess, no matter the variety that is considered. At the same time, also, Pileostigma reticulatum always outperform Gera senegalensis, as I was trying to explain previously. So sometime Pileostigma, sometime Gera senegalensis is similar to the traditional management system. So this let us know that Pileostigma reticulatum is more suited when it's about biofertilization when compared to Gera senegalensis. Next. So this is a slide that showing us the performance of the varieties. So in the Gera senegalensis instance, you see that the best one was Kel. In the Pileostigma reticulatum, the best one still was Kel, and in the traditional management, the best one was Che. But something that we can notice is Leona is always coming back. So Leona is suited for Gera senegalensis, it's suited for Pileostigma reticulatum, and it's suited for traditional management system. We also have Murid, and there is an explanation to this also, because in Chess we experience some drought at a certain moment of the plant growth. So knowing that Murid is very adapted to dry areas, so this is what we expected even before conducting the trial. But we was very happy to see that the new varieties that we have released in 2015 take advantage of the shrub systems. So, however, some varieties that are well appreciated, such as Yasin, they did not take advantage to this management system when you compare them to Leona, Murid, and Che. So these are preliminary results that we will that we have obtained, and this trial will be conducted next year, and we hope we will not encounter the same issues that we had this year, especially the COVID situation, and we will be able to take all the data, and especially the grain yield. So thank you very much.
Thank you, Dr. Dick and Dr. Diangar, for that presentation. We are at the time limit, so we're going to have to forego questions on this one. However, if you put your questions in the Q&A function to the right of your screen on the WOVA platform, the scientists and researchers will be answering the questions for you. They'll be replying in the question and answer format. So feel free to put your questions in there. Even after the presentation is over, just label it by the alphanumeric, in this case, 1B for this presentation. Our next presentation is Science-Driven and Farmer-Oriented Insect Pest Management.
Notre prochaine présentation sera la gestion des insectes nuisibles basée sur la science.
The project is ongoing and is led by Dr. Manuel Tamu, International Institute of Tropical Agriculture, Benin. Presenting today is Dr. Tamo, and the question and answer designation for this project is 1C. That's 1C. With that, we'll go to the presentation.
Good morning, good afternoon, everyone. It is my pleasure to be able to present you some key highlights of our project, science-driven and farmer-oriented insect pest management for cowpea agro-ecosystem in West Africa. So this is a team effort by IITA, MSU, INERA in Burkina Faso, INRAN and University of Maradi in Niger and University of Ilorin in Nigeria. A quick recap of the activities we had scheduled to carry out in FY20. Basically 3 key areas of intervention: biological control with experimental releases of parasitoids, also looking at the behavior of the parasitoids in field host finding capacity, and also assessing factors enabling the establishment of the parasitoids, particularly the interactions with cultivated and wild host plants. The second area was around biopesticides, looking at the efficiency of available new isolates, but also scaling out the production and delivery of the NIMT-BAG technology. And the third area was on capacity development. We had planned exchange visit for technicians and researchers, focus group research, particularly looking at development of educational materials towards scaling of content, and also to expand the farmer interface application as a decision-making tool. Now let's look at what we were actually able to achieve in 2020. So starting with biological control, yes, we were able to carry out the experimental releases of Therophilus javanus in Nigeria as scheduled, but that was a big challenge because of the closure of land borders due to COVID-19. And in the past, we used to basically carry our live parasitoids in the car, drive to the next country and do the releases basically the same day or the day after. That was very simple. These days, with the closure of the land borders, we had to resort to a different type of logistics. And the first one was to use courier services. But here the challenge was that we had to make sure that there were, for instance, direct flights between Cotonou and Lagos, and then that the driver from IITA Ibadan will come down to the facilities of the courier service at the airport in Lagos, pick the shipment, bring it to Ibadan, where our collaborator from Ilorin was waiting with the quarantine people to inspect the shipment and eventually bring it to Ilorin for the releases in the next few days. So you see, it was really not an easy task to carry out, but we actually succeeded and we were very happy about that. I also need to mention again at this point that all our importations and releases of natural enemies are covered by official permits by the regulatory authorities in each of the countries. And you can see here the example, for instance, for Nigeria. Now in Nigeria, we released about 6,000 individuals at 5 different locations around Ilorin, and you can see it here. So this is Ilorin, and this is about 60 kilometers from Ilorin. What is important to note here is that this system of small creeks and small rivers, which provides green vegetation, host plants for the parasitoids and for Maruca during the dry season where no cowpea is cultivated. So this is vital for the establishment and the survival of the parasitoids during the dry season. We also continue to scale out the releases that we had initiated during the initial activity in 2019 in Burkina Faso and Niger. And also here, same problem, the closure of the land borders, but this time the courier services could not assure that there will be a timely delivery of the parasitoid. They would have spent maybe 1 or 2 days in transit in Abidjan. So we had to, this time, looking at commercial air carriers, and the challenge here was to convince them that carrying those live insects would not be a danger, a hazard for them and the passengers in the plane. And eventually we succeeded in that, and so we were able to carry out the releases. We released about 12,000 parasitoids at 7 locations, 4 in Nigeria and Burkina Faso, and 3 in Niger. Quickly, some details of the Niger, Burkina releases here. The same habitats, basically creeks and small rivers. What is important to note is also this big irrigation scheme. So enough moisture and vegetation also during the dry season to assure the survival of the parasitoids. In Niger, we released on cowpea plots around Maradi, but also in an area where we had forests and vegetation, and there was a creek down here, a river down here, which ensured the necessary moisture to keep the vegetation during the dry season. It needs to be noticed, and this is really, we are happy about this, that the parasitoids released in 2019 with the initial activities was recovered from the field in both countries, although still from few locations, but this is a really encouraging news and we are on the right path. So also during the initial activities, we had started with initial colony rearing of the parasitoids in both countries, and this now has been expanded in 2020. And actually in Burkina Faso, we were able to do some releases using parasitoids produced locally from the INERA labs. And then we wanted to look at the host finding behavior of the parasitoids, and I'm sure you remember Cynthia on this picture that was taken in 2016 when we had the official releases, the first official releases of the parasitoids in Benin. And we used for that purpose these big cages, and which were quite useful and gave good results in terms of measuring parasitism. And we thought would be using the same approach now to study the host finding behavior of the parasitoids. It has been used for some parasitoids with success, but unfortunately our parasitoid was a bit peculiar because once released in the cages, it just basically stayed on the walls and didn't move for many hours. Maybe attracted by some static charges or color, the texture, we don't know exactly. Of course, the parasitoid eventually moved down to the plant and parasitized Maruca, but not very useful if you need to do very detailed observations and time them. So these cages can still be used for quantitative parasitization tests, but for the host finding behavior, we had to resort to different methodology. And that was actually not too difficult because, as we found out, our parasitoid is a very docile animal. So once released in the field on a cowpea plant, it doesn't really move too much, and you can observe it very easily. And we had captured that now in a video, which I'm going to show you. And so basically, let me quickly stop here and show you some details. So this is the ovipositor, the slender one, and these are the ovipositor sheets where the ovipositor rests when it's not used. And as you can see here, the parasitoid is using the ovipositor to drill through the flower. And this is kind of a breakthrough observation. We didn't know about that until we really noticed that really in the field, because our lab studies were not using flowers, were using just a larvae, and we thought the parasitoids would also just parasitize larvae in the open space. But here now we have the proof that the parasitoids can really drill through the flower, find a suitable larvae and parasitize it, as you can see from here. So this, we are really very happy to have done this experience and to have made this movie. With regard to biopesticides, we have focused our work on this pest here, the pot-sucking bug Clavigralla tomentosicollis, and this is what happens to the same guy if it comes too close to our biopesticide, an entomopathogenic fungus called Beauveria bassiana. Just looking at the example of Niger, we have compared different biopesticides with a chemical insecticide and untreated control in their ability to reduce pot bug populations using both a susceptible local variety and an improved variety which is a little bit more field tolerant. So if we look at the population levels in the untreated control, the population is quite high and substantially higher as all the other pesticide treatments, and comparable results have been obtained also on the tolerant variety. And this is also confirmed by yields. And if you look at the yields of the untreated control is much lower than generally the trends of the yields in the pesticide treatments. Now, another important activity that we already started in the initial activity phase was the scaling of the neem tea bag in Niger. And so this is a picture we took during that time. And basically our team in Niger has done a great job in providing a game changer, these easily transportable motorized mills to grind the neem grains, because this is really a big time saver and also can reduce the drudgery of the women involved in the community-based production units. So an enormous improvement over the traditional method of using the mortar to grind the neem grains. As I speak now, we have probably produced over 3,000 neem tea bags, of which 2/3 have already been sold. Over to capacity development, and this unfortunately is the activity which has been impacted most severely by COVID-19. For instance, group training for the neem tea bag production had to be reduced substantially, and only 6 participants could take part. All of them were women from the 3 production units. Training topics included the identification of insect pests, how to fight those pests with nature-based solutions, of course, including the neem tea bag, how to prepare it, how to apply it in the field, and then some basic skills in agriculture entrepreneurship, and also basic skills how to assemble, operate, and maintain those mechanized mills. Other capacity development activities included degree-related training, so we were able to recruit 1 PhD and 2 MSc students in both Burkina Faso and Niger, all female. The farmer interface application has been expanded to include a scouting protocol with voice commands as part of the cross-cutting activity. And also an unexpected but very welcome addition was to start to produce a new Sabo animation on the production and delivery of neem tea bags. And this is very important because its educational contents can be translated in different languages with a potential impact beyond Niger. So the lessons learned in 2020, we have been able to carry out the releases of biocontrol agents thanks to improved logistics in spite of COVID-19. We had some unexpected challenges in the behavioral studies, but we have been able to mitigate that by in-field observations, and now they will be complemented by chemical ecology studies in the next season. The good news that our parasitoids is established at few locations in Burkina Faso and Niger after the initial releases. And the game changer, our small mechanization units, which can really enormously improve the output of the women-led community production units for the neem tea bags. And unfortunately, the major impact of COVID-19 on our capacity development and socio-economic activities. So like the exchange visit had to be canceled altogether. The focus group research is postponed. The group training, as you have seen, has been enormously reduced. So at this point, I would like to thank sincerely USAID through the Feed the Future Innovation Lab for Legume System Research at MSU for supporting our work. And also I wanted to thank my IPM team on the ground for really sticking to the work during this difficult time. And thanks to all of you for listening.
Thank you, Dr. Tamo, for that presentation. We have time maybe for one question. I don't see any questions in the question and answer function, so I'll ask one of my own. Dr. Tamo, could you join us for a moment and
yes,
a brief question? So for our Southern African colleagues, how exportable are these technologies to Southern Africa? Would they be applicable there as well?
well, maybe the parasite, can you hear me first?
Yes.
All right. Yeah, parasitoids, yes, that shouldn't be a big problem. In fact, we are already exploring that possibility because Maruca can be a big problem around Mozambique, for instance. Mm-hmm. But the bigger problem is probably with neem because as far as I know, there is no neem growing in Southern Africa. There is a kind of a cousin of neem, which is Melia azaderac, but it's probably not exactly the same ingredients. So we can look into that, definitely, but it cannot be exportable from one day to the other. Let's put it like that.
Perfect. Thank you very much. Well, that's all we have time for on this session. Thank you very much, Mano. That was a great presentation. And
thank you.
Concludes our question and answer session. This also concludes today's public session of the Legume Systems Research Innovation Lab's Virtual Convening 2021, Day 1. If you have not yet participated in today's poll to vote for your favorite legume, please take a moment to do so. And to review any of today's pre-recorded presentations, click on the Research tab on the Whova page. Also check out the community feature where you can engage with other attendees and find additional resource materials. I also think that I want to remind you that if you have other questions that occur to you, you can always add them into the question and answer function. Just identify it by the session 1A, 1B, or 1C. And with that, I will turn it over to Jan. Thank you.
Thank you, John, and also to all of our researchers for those very interesting and
merci à tous nos chercheurs.
So, as John mentioned, please continue to use our session Q&A feature, and we look forward to having everyone join us again tomorrow for our public session, which begins at 10 a.m. Eastern Standard Time. Also, as a reminder to our project affiliated attendees, the continuing closed program session will begin at 12 noon Eastern Standard Time today. So for you, there is a short break, and for our public session attendees, we look forward to seeing you tomorrow. Thank you and have a great day.